Welcome, Land Stewards. I'm Rachel Worling with the OSU Land Steward program, and it's my pleasure today to introduce Scott Goode, who's going to give us an introduction to soils for the Land Steward training for the soils module. So I want to tell you a little bit about Scott before we get going. Scott Goode is an environmental scientist who has worked in water treatment, bioremediation, and research in the role of environmental factors in soil chemistry. He lives in the Rogue Valley and is currently a research scientist with the University of Nevada's Desert Research Institute in Reno, Nevada. He's a longtime land steward volunteer, and we're very grateful for this presentation. All right, Scott, take it away.
Right. Thank you, Rachel. So this we're going to look at soils from the perspective of what constitutes a really healthy soil. There's a lot of videos out there on various aspects of soil biology and soil management and so forth, but here we're just going to really take a close look at what constitutes a really healthy soil. So soil is formed from a relationship between climate and geology and biology, and those are the principal forming factors in soil. And this is usually put into the acronym C-SPOT, stands for climate, slope, parent material, organisms, and time. And varying any one of these will vary the rate at which soils can either improve or develop or deteriorate. And in the sense of climate, in a very, very moist environment, you'll get much faster soil development than you will in a desert. On a slope, you're going to have the process of every time you start forming a soil, it has a tendency to wash down the slope and into a valley. In parent material, the soils that form from a granite are very, very different than ones that form from, say, a basalt, because the minerals in each of those are different, and it's the minerals from those that produce the mineral side of a soil. And organisms really are of critical importance in the formation of a soil. In a desert where you have absolutely no biomass accumulating on top of the soil stays the same for centuries and even millennia. On the other hand, if you have rich old growth forest, the materials that fall on that generate very, very deep, very, very rich soils. And so given enough time, even relatively unvegetated area will eventually accumulate enough organic matter in it that it will begin this soil forming process. So you can see here in this, you have the stage one where you end up with a bedrock, whether that's from a landslide or a volcanic ash flow, or shortly after that, organic matter begins to form on it in the form of lichens. And the lichens and the fungi and bacteria in a lichen begin to, the fungi will begin to put out organic acids and dissolve even solid rock. And the photosynthesis that goes on in the bacteria in the lichen will generate biomass and eventually accumulate even a thin layer of organic matter on the soil. And that thin layer will provide the opportunity for other plants to begin forming. So there you see on section 3, where soil horizons form, you begin to get the separation of an organic layer at the surface. And then what we think of as a soil layer in the A horizon, where you now have something that looks like a soil and not just an accumulation of rock. And then eventually, as the soils begin to mature, that A horizon begins to separate out, and you end up with organic rich material in the A horizon and more clay and mineral materials in the B horizon. And this is just the sort of the normal progression of the way soil develops. Now, the other important part of that is that vegetation layer. When you start out with bare soil, there are bacteria that will begin to grow in the soil. And so most of the biology that is in there is bacteria and stuff that eats bacteria, like nematodes and arthropods and that sort of thing. With a little time, the weeds and the grasses begin to form, and fungi the fungi that are usually associated with the roots of these weeds and grasses become a component of the biology in the soil. And as that progression goes from weeds to mixed herbaceous plants to shrubs and then to deciduous trees, you can see there that the fungal to bacterial ratio changes, that there gets to be more and more fungus and generally less and less bacteria as it goes toward a climax forest. And so in the process of moving a soil along, like say a soil that you want to cultivate, if you are interested in growing, say, vegetables in that soil, then you'd be looking at moving that soil along to where it gets to the layer, the point where it has a fungal to bacterial ratio of 0.75 or 1 or something like that. And if you're moving toward an orchard, then you're going to want to have a fungal to bacterial ratio of above 2 to 1. And there are ways that we can move that soil biology just by the cultivation techniques that we use, the plants, like for instance, cover crops that we grow in it, to begin to move that progression along this biological evolution in the soil. Now, we were talking about the slope has a lot to do with soils. This happens to be a cross section of the Rogue Valley where I live, and on the west side of our valley we have rocks that are thrown up from a subduction zone, and they're very, very metamorphosed and to some degree volcanic rocks. And on the east side of the valley we have the Cascade Mountains, which are made up of a very different kind of geology, granodiorite, so that is the difference between the parent materials that generate different soils on opposite side of the valley. And on the steep slopes, you're going to have soils that have a lot of rocky chunks in it. That's what colluvium is rocky chunks that roll down the hill and erode a little bit as they do it. And then down in the valley, in the terraces of the valley, the accumulated sands and silts that wash down from the hill slopes begin to develop soils that are richer in sand, silts, and clays. And the clays get there because every time you have a flood in one of these valleys, that floody, muddy water is rushing down up onto the floodplain. And when it gets out on the floodplain, the speed of the water slows down, and the clays will settle out. And so you'll often find clays not in the Central River Valley, but up on the terraces on either side of the valley. And then down in the valley floor where you see Central Point and Medford soils, those are both really wonderful silts, loams that are have silts and often sands in them as well. And the reason that is because as floods come through, they will actually wash the clays out of those soils immediately adjacent to a stream, and you'll end up with silts in those river valleys. And that's why the very famous soils of the Tigris and Euphrates Valley or the Nile or Mississippi are so rich, because they mix soils from upstream and alongside, and then accumulate richer and richer loams as time goes on. So let's look at really what a healthy soil looks like. Now, To find a healthy soil, you might need to go to some place that's never seen tillage, a nice rich forest floor, for instance. And if you look at those soils, they're the most productive soils. And so these are what we would really want to aspire to in developing our soils over time. And as you can see, 40% of it are the mineral grains themselves. These will be sand, silts, or clays. And those provide what we call the texture of the soil. That means the ratio of sand, silt, and clay in the soil. And then you have the organisms that are growing in the soil. And they're very critical. And the more of them there are, generally, the healthier the soil is. And then you have the roots, and then the decomposed materials that come from those organisms and plant detritus. And that generates, what we really, really look for in a healthy soil, humus. And then the other part of the soil are the open pores in the soil and the moisture that's in the soil. Now, that moisture we call a soil solution because it's not water. It's full of biological or biochemicals that are generated by the life in the soil. It contains dissolved minerals that are in the soil. And of course, as you irrigate or get precipitation in the soil solution begins to fill those open pores. And then as your soil begins to dry out, the percentage of the soil that are open pores increases. But that open pore space is an incredibly important part of the soil, first of all, for moisture storage. But even more important is, as we'll see, that actually is the volume of the soil where most of the life actually lives, in the pores of the soil, in a healthy soil. So we were talking about soil texture. That's the ratio of sand, silt, and clay in a soil. And you can see the relative sizes here. Sand grains you can easily see and easily feel. Silt grains will feel sort of like flour in your hand. It's fine, but you can still just barely feel the grains. And clays are so fine they're just a very, very fine powder in the soils. Now, the remarkable thing is that once we start looking at the life in the soil, we realize that there are bacteria all in the soil. They're 10 times smaller than the clay particle. And so what that means is that on top of a clay particle in a good, healthy soil, you will have fungi and bacteria climbing all over the clay particles, the silt particles, and the sand particles. So relative to even a clay, the biology gets even finer than that. So the finest part of the clay soil, the clays, not all clays are the same. The clays are completely a function of the parent material that the soils have formed from. So in the upper left, you have these nice kind of cornflake looking clays. These are chlorites. These are the clays that we have in on the West Coast, where we have metamorphic rocks from the subduction zones. And they're very, very well behaved. They have a nice surface area, which means that they can attract and hold on to nutrients really pretty well, because there's lots of little crevices in there. And then below that, you see this is a soil that forms or composes a great deal of the Midwest. And they're well-behaved soils in that they don't shrink and swell. But at the same time, you see they're very, very smooth, and their surface area is noticeably smaller than some of the other clays. And as a result, they can't hold on to nutrients quite as well as some of the other clays. And then up in the upper right, the stuff that looks like phyllo dough or puff pastry, perhaps, this is kaolinite. This is a clay that comes out of granites for the most part. And you can see it has huge surface area. It's just tiny, tons of little flakes there. And if you've ever worked with porcelain, this is the clay that forms porcelain. And when porcelain gets wet, or it'll turn into a soup. And that's because each one of these little flakes will begin to come apart, and you'll end up with this suspension of all these little flakes. And that forms the slip that is used in pouring into molds for forming porcelain. Now, this bottom clay is the one that gives all of the clays a bad reputation. Now, right now, we're seeing it when it's really, really dry. And so if you've ever taken a balloon and sucked all the air out of it, and it gets all this weird crinkly look, well, that's exactly what's happening here. This soil is what locally and a lot of places they call the black sticky, because it's very, very sticky. It shrinks when it gets dry, it swells when it gets big, and as a result, when it dries out, it's really recognizable by all the cracks and polygons that it forms when it dries out. Now, it's true, this does have a huge amount of surface area, but one of the characteristics of it is that when moisture actually moves into the interior of this clay, the clay holds onto it so hard that often the moisture can't make it out of the clay and into plant roots. So this is a very challenging clay, but a lot of clays are actually really wonderful and can hold a lot of nutrients in the soil. So when we look at a clay, in that last picture, you can see, particularly with that kaolinite, that it's made of these layers. All clays are made of layers. They're just, some of them are more obvious under a microscope. And those layers have little positive charges on each of the surfaces of each one of those layers. And that slight negative charge will attract the nutrients and dissolved minerals that are out here in the soil solution, and it will draw them in and store them. And this is what we call a cation exchange complex. All of these positively charged ions are called cations because they're positively charged. And most of them are nutrients that plants need. And so you can think about clays as this sort of parking garage where all this nutrients can go and be held into the soil. And as long as they're not too dried out and the right kind of clays, then they can, as soon as the plant roots come in proximity to them, they can draw out those nutrients as they're needed. So this is really the microscopic or nanoscopic view of the way clay particles operate in their relationship, particularly with soil nutrients. Now, when you, there are a number of ways of finding out what your soil texture is. On most soil reports, you can actually get the lab to do this test and tell you whether or not you have or what the texture of your soil is, depending on what the ratios of sand, silt, and clay are. The significance of, or why it makes a difference what your soil texture is, that sandy soils react very, very differently to precipitation and irrigation than clay soils do. As you might expect, if you take a piece of drip tape and irrigate, or a trench of irrigation trench, in a sandy soil, the water will move very, very rapidly down into the soil, so that after an hour, it's down to say 48 inches, and that's about as deep as most plant roots will go. So if you let your irrigation run longer than an hour, all it's going to do is leach out the nutrients out of the soil and put them down into the water table. On the other hand, and the other thing is that it won't transmit the moisture very far away from the irrigation source. So regardless of how long you let the irrigation run, it's not going to get any more than a foot away from the irrigation source, regardless of how long it runs. And so with a clay soil, on the other hand, clays will transmit water much more effectively. So as you let the irrigation run, you can see it will carry it out away from the irrigation source, a good 2 feet out away from the irrigation source fairly quickly. But it won't sink down into the soil nearly as fast. And so as a result, if you have a sandy soil, you're going to want to run your irrigation for a much shorter period of time and much more often. And with a clay soil, you're going to want to run the irrigation for a longer period of time, and that will allow you to go longer between irrigation. So that's just one example of where your soil texture, knowing your soil texture can give you a lot of information on soil management. So now let's take a little, speaking of soil moisture, let's take a little closer look at the soil solution and what role it plays in the soil. So here it is. It's the transfer media for nutrients that are locked up in the clays out into the soils, and allows that's where particularly plant roots, not necessarily fungi, but plant roots, that's the place where they can get have access to the nutrients in the soil. Now, a plant root by itself is remarkably inefficient at acquiring these nutrients. And as a matter of fact, without any biology in the soil to help it, the plant root has to use some of the carbon that it's taken in from the atmosphere that it would normally use for developing tissues and sugars and fruit and so forth. And instead, it has to use that to create a very weak organic acid to put out into the soil solution around it, and hope that some of the particles that come from that soil exudate will migrate across through the soil solution and tickle that soil particle, that clay particle, and wiggle loose a nutrient, and then it would have to float back across by osmotic pressure mainly, back to the root, and then go to be taken into the plant. Well, when the plant puts off this hydrogen ion into the water to knock off a nutrient, it has no idea and no control over what nutrient it's going to get back. And so you can see this is a remarkably inefficient system of nutrient acquisition by the plant. Not only that, since the distance between the plant and the particle is not very far, this process can't extend very far into the soil, very rapidly in the soil area right around the root, it will deplete the nutrients that it actually needs in order to grow. And so in traditional agriculture, the way that because there's very little life in traditional agricultural soils, then the only way to replace those nutrients is to add chemical nutrients into this. And when you do that, it completely fills the soil with those nutrients just in order to refill that little gap in between there. And so the rest of the nutrients that are put into it remain out of touch, but are readily available to leaching by the soil solution. So when you have a soil analysis done and you look at the chemistry of the soil, what you're looking at is not the minerals in the soil itself. You're only looking at the nutrients that are actually dissolved in that soil solution. So what this is telling you is not what is in your soil, it's what's in the soil solution, the liquid part of the soil. So a very famous soil scientist went out and he was wondering, well, how many nutrients are actually in an average soil? And he went through and looked at 3 million different soil samples from around the world. And what he realized was that in the soil itself, in the soil itself, there are plenty of nutrients. The only problem is they're locked up in the minerals in the soil. And if you look at the difference between what's actually in the soil and what the plant's needs are, there's really, if there was just a way of getting to those nutrients, there would be absolutely no reason why you would need to amend the soil. And so that's really the key to good soil health, is making these nutrients available to the plants that you're trying to grow. And so here that you see this depletion zone immediately around the plant. And that's the only part of the soil solution that it can reach. So that brings up the question, why would plant roots and root hairs have evolved in such a limited reach into the soil? And the answer to that question is soil organisms. Plant roots actually are there to support soil organisms. They're not there necessarily. I mean, they can, but they're not very efficient at acquiring nutrients themselves. But as we'll see, they're actually pretty good at providing a base or a substrate for organisms who can assist in that process to thrive. So when we look at a good, rich soil ecology, this simply represents the ratio of critters in the soil. They are certainly not stacked in layers in the soil, but this gives you a sense of the relative numbers of them. And you can see these enormous number of bacteria and actinomycetes are there that, well, remember, those are small enough that they can actually live on clay particles. And so that's why there are so many of them, is because there's so much substrate for them to live on. And so Elaine Ingham, over a decade ago, sketched out really for the first time how this soil biology works, and described it as the soil food web. And that is that all of the different parts of the soil are interactive with each other, and that's the system that's actually, or that's one way of looking at the soil biology, is in trophic levels. A trophic level means that in the first trophic level, all of the energy is coming from the sun and from the plants themselves, utilizing sun and carbon dioxide. So that's the bottom level, and that generates organic matter. And then the organic matter feeds the second trophic level, bacteria, fungi, and nematodes. And then they are the food for the third trophic level, which are in turn the food for the fourth trophic level, and food for the fifth trophic level, which is where we, the part of the trophic level that we occupy, because we generally eat parts of those plants. But it turns out that this is just a way of sort of organizing who's who and kind of, what they're dependent on. The actual soil itself is organized into zones or regions or spheres. And at the surface, you have biomass coming in into the soil, and that's detritus. And there's a whole host of critters that do nothing but live in the detritosphere, break it down, and make it ready for the next layer in the soil. And that layer is the drillosphere, and it's full of a whole bunch of critters, notably worms, that then take those nutrients from the detritosphere and take them out into the soil and distribute them all over through the soil. And then if you think about it, in all those little pore spaces that are in between the soils, there's a whole nother ecology in there, that some of them that are aquatic because they're living in the soil solution, and some of them are living on the surfaces of the soil aggregates themselves. And we call that zone the porosphere. And then if you start looking microscopically down on the surfaces of the soil, you have all the critters that are living actually on the soil, those bacteria and fungi. And they are able to actually drill into the minerals, into the soil minerals, and dissolve off of the soil surface, because they can secrete enzymes and organic acids that can actually dissolve those minerals and leach out or remove the nutrients that are in those minerals and make them available to the whole rest of the soil volume. And then the final layer, the one that's really kind of received the most attention, is called the rhizosphere. Rhizo meaning roots, and the area around the roots. And there is a cascade of critters that are absolutely dependent on a living root being adjacent to them for nutrients and so forth, and that this layer of biology can generate, turn both the nutrients around them, but also generate biochemicals that in fact the plant takes in and utilizes. For instance, auxin, which is an important enzyme in plant growth, much of the auxin is actually produced by biology, by microbes that exist outside the root and then transmit that auxin into the root itself. And so this zone out around the roots is this beautiful, chaotic region that is probably the most active part of the soil. And if you look at the effect of a root going into the soil, a plant germinating into a soil. So here you have a soil that's just soil before a plant germinates. But the minute that plant germinates and its roots started going in the soil, it's taking carbon dioxide and sunlight and turning it into sugars and feeding that to the microbes that are living right around it. And so immediately the whole soil structure around it begins to take on a characteristic that's driven entirely by the plant roots. And the longer the plant lives, the more it affects the soil biology around it, generating this rhizosphere. And the relationships that exist between these individual species in the soil, between them and each other, are symbiotic. And of course, they're symbiotic with the plant because the plant is putting out nutrients that keep them around. And so you can see that this is a very, very specific biology, and there are very, very specific critters that live in it. And so 50% or up to 50% of the sugars that are generated by a plant may well be expended just trying to feed the biology around it. Well, the healthier the soil is, the more nutrient rich it is, then the less of this the sugars and proteins that it generates does it have to expend on acquiring nutrients, and the more that it can spend on growing leaves and branches and fruit. And so that's another reason why having very, very healthy, optimal biology in the soils can allow the nutrients to remain in the plant and not try to grow soil biology. Now, the number of things that the interactions between the plant and the biology around it are incredibly complicated. The more we look, the more we see. And so it is the interaction between these, the symbiosis between these, that is the basis of good, healthy plant growth. So one of the most famous of these are the mycorrhizae fungi. They're getting a lot of notice over the last decade or so. There are two forms of it, the endomycorrhizae, which actually these fungi were among the very, very first soil life forms to occur as plants began to emerge on land. And very much like a lichen, these ectomycorrhizae have this intimate relationship with the plant. And in fact, the very root, if you will, of the fungi grow inside cells on the inside of the root of the plant. And this provides the fungi protection from the world outside, but it also allows the fungi to communicate directly with the biology on the inside of the root. So if the plant needs something, it can communicate it to the fungi, and then the fungi has a capacity to look all in the soil around it and find that nutrient. Now, later on, fungi, ectomycorrhizae fungi began to, or evolved, and they form a fuzzy layer all over the outside of the root. And so now, instead of the plant protecting the fungi are capable of protecting the plant. And we'll see an example of this later. So here's an illustration of mycorrhizae versus root. The little yellow spindly things in here are actually the seedling's roots. And this entire cascade of white fuzzy hyphae and mycelium going out into the soil are able to reach into places into the soil grains themselves and pull out nutrients in response to the plant's needs. And that's because the filaments of these things are absolutely nanoscale. They're so small they can actually get into the crevices of a clay particle. Another famous member of the rhizosphere are the rhizobium bacteria that grow on legumes and other plants, famously on legumes. And these nodules are able to convert nitrogen out of the atmosphere and out of the soil atmosphere and pull the nitrogen out and make it immediately available to the plant. And a remarkable innovation of symbiosis that has had profound effect on the evolution of plants. Now, the beautiful thing about this system is that not only do the mycorrhizae cover and feed and protect an individual plant, they are able to connect the plant roots between two different kinds of plants, which in fact specialize. Monocots are really, really good at being able to metabolize and make a phosphorus available and generate lots and lots of sugar to keep everybody going. Legumes, on the other hand, are really, really good at generating a surplus of nitrogen. And that process of nitrogen development requires a great deal of phosphorus. And so the monocot provides the phosphorus, which allows the rhizobium to generate the nitrogen, which then the mycorrhizae carry back over to the monocot so that it can produce more phosphorus. And you see how this goes. Plants don't compete with each other. Plants are actually sharing in this beautiful soil food web. And that allows them to specialize. So depending on the depth that their roots go and the kind of roots that they have and the critters that grow on those roots, each individual plant can specialize in a particular kind of nutrient or get down into deep moisture. And then they work together as a guild to share those nutrients. And that occurs in a healthy environment. And that's why growing cover crops and companion planting and those strategies in food production are so beneficial, because it allows for the interaction of different plants in order to share nutrients. Now you see the one oddball here on the end, turf grass. You see it has really no roots whatsoever. That was because it was bred entirely to feed off of applied fertilizer. So that's the answer to why a root isn't really that good at pulling nutrients out of the soil. A plant is really, really good at feeding biology and microbes that do that work for it. And so even after the root is gone, it provides space in the soil for new roots to come along and increase the soil organic matter by adding to the detritus sphere up here. And then also producing lots and lots of pore space in there for critters in the soil to occupy, including the roots of other plants. And so since each one of these parts of the soil, the different spheres of the soil interact with each other, that is really what provides for a healthy soil. Well, if you till a soil, you destroy this entire biological network, all these relationships, and of course you kill off a lot of the critters that are in there. The other thing that you do is that you introduce oxygen into the system. And when, for instance, you plow in the detritus from above and mix it with oxygen and bring it down into the soil, what happens is that oxygen will oxidize and provide for these very, very aerobic species that are pretty good at tearing apart the biomass. But what they do is they pull out the nutrients, they feed it to the plant, and the carbon, the organic matter that compose that detritus is used up. That biology in its metabolism, the byproduct of it is carbon dioxide. So in a system where you have tillage, bringing oxygen into the soil and bringing biomass down to the soil, it will outgas carbon dioxide, a process called priming, much, much, much higher than if you use no-till systems that allow for each one of these different spheres to remain intact. So here we have the detritus sphere at the surface. And in the detritus sphere, you have all these arthropods and worms, a whole specific group of worms that are the shredders. And what they do is they shred this biomass, they turn it into fecal pellets, all different kinds of fecal pellets. And then the mycorrhizae that are growing around roots are available to that. And in fact, those fungi will grow up into the detritus. And as it's being decomposed by the detritivores, it can pick that nutrient up and carry it directly to the plant. So the detritus sphere is such a critical part of the soil because that's where the nutrients are cycled. And if it's efficient in that a majority of the detritus that comes from a dying plant or a dead plant is cycled back into the system, then the nutrients, it really never loses any nutrients. And you're having new nutrients added from the bacteria digesting the soil aggregates. And so the soil gets richer and richer and richer. And so right now, there's really very, very little detritus sphere left on the planet. It's all been plowed or cleared. And the only place that you really find an abundance of the critters that used to make up the detritus sphere are in compost. And that's basically what we do. We replace the detritus sphere by taking all the biomass off someplace and putting it into a pile and letting these guys do their job and then picking that up and trying to feed it back to the plant. So we're sort of taking on the role of many of the other spheres of the plant and trying to do all that work ourselves. It's a lot of work. So as the detritosphere generates these nutrients and they become available, you have a whole different cascade of levels of drillovores. That drillo is the Greek term that means anything that resides in the soil. That goes for everything from snakes to worms, but in this case, we're really looking at the snakes. So you have epigeic, pardon me, at the worms. You have epigeic worms that live their entire lives up here in the leaf litter. They never go underground. All they do is eat and poo, and right up at the surface. And then you have the anessic worms that come to the surface. They feed on the detritus. They carry that back down the hole, and then they feed on the minerals that have been generated by the biology in the deeper in the soil. They mix those together and they bring those up and leave them as casts, the mixture of those two, onto the surface, producing incredibly rich soil all the time. And as long as that's not disturbed, the soil just gets richer and richer and richer. And then you have endogeic worms that take material from the surface or just below the surface and from that's accumulating in the channels here, and they work it into the soil all the way through the soil, filling it full of passages that moisture and nutrients and roots can penetrate. And then there's one other group, remarkable. These are the geophagious worms and other critters, and they actually eat rock. They're able to go in, excrete digestive juices into the soil, ingest those nutrients, and then leave those as either a biofilm or exude those by either as a biofilm on the channels of the pore spaces, or provide them as fecal material that other critters come along and eat. So here you can see this relationship, moisture making it into the soil through the pores. Every single one of the anessic channels is coated with a biofilm that's full of calcium nodules and mucus that holds onto nutrients and moisture. And then they put their casts in there and bring middens up to the surface. So you can see that this is an incredibly critical part of good soil health. And so then we look at the porosphere that's in between all the channels here in the soil. And so here you have an open volume. In this case, it happens to be filled with moisture. And so you have nematodes swimming around in it, and ciliates and bacteria and fungi and plant roots coming into it, and larger, more megafaunal critters like mites and springtails and arthropods and so forth. But if you think about it, these zones inside in the pore space are like little tidal pools or oases that are full of biology and keep the nutrients cycling through the soil. And once again, if this is disturbed by tillage, all this goes away and all you have is dead soil. So one of the critical things in these soils are actinomycetes. They form these beautiful thread-like, net-like layers that will, here you can see these are soil particles, and you can see that they're being completely networked and covered over by actinomycetes. Well, these threads are actually pretty strong. And so what they do is they allow individual clay-size particles to be drawn together in a network and form as larger and larger aggregates. And the other thing about actinomycetes is that they are also nitrogen producers. They're able to pull nitrogen out of the environment and feed them to plants other than legumes. Famously, alders in very, very poor, very, very low pH soils always have actinomycetes completely growing over their roots. And the only way that they can get any nitrogen is for the actinomycetes to take the nitric acid out of rainwater and take that nitrogen, convert it into a plant available form, and feed it to plants. Protozoa are working their way constantly through the soil, digesting one thing, putting out fecal material and biofilms. And these guys slather every single surface that they crawl over with biofilms that hold nutrients and enzymes and often plant materials that actually can help stimulate root growth. Then you have the nematodes. That fill the soil. They are really one of the things that keeps the populations in balance in the soils. You have all of these bacteria constantly breeding, and there needs to be something that comes along and grazes on those and turns them into fecal material that then in turn becomes a nutrient for another layer, another trophic layer. And there's all kinds of nematodes, and you can tell one from another generally by their mouth parts. This one has these little tentacles that go out there, and they're great at gobbling up bacteria and ingesting them. But there's all different kinds of mouthpieces on these nematodes depending on what they do. And you can just imagine what this guy down here, Mr. G down here, what he must go after. Pretty remarkable. Now, this is the nematode that gives nematodes a bad name. You see there's a spike, a harpoon right here in its mouthpiece. And what it does is it launches that off into a plant root, and then it begins to draw off the nutrients out of the plant without really providing anything in exchange. So this is the nematode that really people are often worried about. But it's only one of dozens of other nematodes. And most of the others are providing a valuable surface in the soil. Now, here's the amazing thing. Remember we were talking about you have fungi, the ectorhizal fungi coating the outside of plant roots. Well, one of these fungi lays in wait for one of these plant-eating nematodes to slither along, and it reaches out and lassos it and then digests it and then feeds the nutrients from the nematode to the plant that it was about to attack. So let's take a look at this aggregatosphere, hard to say, but it's all the critters that are actually living on the mineral grains themselves. And like I said, the fungi are able to secrete organic acids, oxalic acid notably, that actually dissolve soils, and they're able to actually drill into a mineral grain and then invite bacteria into the drill hole. And between the two of them, they'll actually extract brand new nutrients out of the soil grains and bring them into the soil and the soil solution. And so every single season that goes through that you have a healthy soil, these guys are adding nutrients to it every single season. And so if you have these guys operating effectively, and you have the guys in the detritosphere happening effectively, working effectively, cycling the nutrients into the soil, every single season, the soil just gets richer and more full of nutrients. So you can see these bacteria, you can see the surface of this aggregate is completely covered over by these critters. And here you can see the background. These are the bacteria that I was saying, that 10 times smaller than a clay particle, and you can hear some of them actually drilling their way into a mineral particle. Oh, the other important thing about this is that as they're doing all this activity, bacteria don't have a mouth. They don't, gobble on something and then ingest it and then expel fecal pellet. What bacteria do is they essentially slime everything that they crawl over with enzymes, and then whatever dissolves that they can eat, they'll ingest, and they leave the rest of that enzyme-laden biofilm covering all of the mineral aggregates. And so over the course of time, in a healthy soil, if you look at the aggregates, the actual grains of soil, over the course of time, you won't see sand or silt or clay. You'll see sand and silt and clay completely covered in a biofilm. And these biofilms are glues that will hold an aggregate together, but they're not water soluble. And so when these aggregates get wet, they don't fall apart. And so that develops a really good soil structure. See, these biofilms are completely covering these grains, and that develops this aggregate stability. So with all of these things working together, the actinomycetes causing these networks, the bacteria and so forth generating the biofilms, gluing all these aggregates together, holding larger aggregates together, providing spaces for holding soil moisture. This is and forming up these aggregates that are able to hold together even when it's wet. That's aggregate stability. So I think everybody recognizes that humus is a good thing, and that's what we're really looking for in a good, healthy soil. And of course, humus is basically decomposed organic material, or the fecal material or biofilms that are put out by soil biology. And for a really healthy soil, looking at a minimum 3% organic matter, and really up to around 5 or 10% organic matter is what demonstrates a really healthy soil. So if you look at, just like we looked at the clay grains through electron microscope, if you look at the humus through an electron microscope, this is what you see. It's just this fluffy, if you could see it in color, it'd be dark brown or even black. And this is just the fluff and duff and fecal material and biofilms that are left over after biology has its way with the soil. So in the detritosphere, you're breaking down all of the biomass that's in that it makes its way into the detritosphere. And the sugars and the carbohydrates are gobbled up almost immediately. They're used for the nutrients to keep the whole system going. But there are these other more resistant organic components to plants. Lignins are what give wood its strength and can provide make wood resistant to decay. Chitin is what forms the watertight surfaces and provides strength in tissues. Chitin is the same material that crab shells are made out of. So it's very tough, very strong, and very flexible. Pectin is kind of the brick and mortar of plants. It holds the plants together and provides their structure. And then flavonoids carry on all sorts of metabolic processes in the soil. And subrin is, if you really want to look at subrin, look at the outside of a potato. You know, that brown, rough, waterproof, resistant layer that's on the outside of a potato? Well, that's subrin. and it's made up of these chains of fatty acids. So each one of these, you see these ring compounds that form up these molecules, these biological molecules. Well, these are very strong structures. And so as the detritivores come and shred everything, as long as there's not disturbance like tillage or oxygen being introduced in the soil, then these are the components that eventually form resistant organic soil organic matter in the soil. So this is what forms the humus that actually stays around for a while. And so each one of these individual fragments are floating around, all different kinds. And if you look into the soil, you find these chunks floating around in the soil, but not in the soil, in the detritosphere. It can happen in the soil, but more effectively in the detritosphere. Well, it turns out that all of these have these little red nubbins on the end. Well, those are the negatively charged particles that allow humus to have such good nutrient holding capacity. The only problem is that they put a negative charge around this guy and a negative charge around this guy and a negative charge around this guy. And as a result, they repel each other. Well, it turns out that as you decompose organic, the biomass, remember there's calcium and magnesium and all these wonderful nutrients in those cells, and they end up forming little molecules or atoms really of calcium and magnesium and other things that are floating around in this biomass. And as long as the oxygen doesn't get to them to oxidize them, to turn them into a salt basically, then these atoms, they're called free radicals, they are positively charged, and they can reach out and pull this particle and this particle and this particle all attracted into the same area. And eventually they'll grow up to form these huge, fluffy humic micelles or molecules, macromolecules that form humus in the soil. And once they're formed up, that once again, they have a negative charge on their surfaces. And so you see all these little molecules that are attached to the surface. The little red and white ones are water, and then the purple ones are, in this case, calcium, but they can be any positively charged nutrient. And the humic molecule will hold on to them just strong enough to keep them out of the soil solution, but incredibly available to plants that are growing in it. And so this is the process of the formation of stable organic matter and humus, that we think on humus as humus. And you can see that it's this dance between nutrients that once composed part of plant tissues being liberated out into the detritosphere, and then they can draw these other organic molecules together, forming humus. And these systems are scattered all the way through either the detritosphere or the soil, and they begin to form more and more and more complex systems of molecules that are great at holding moisture and nutrients. Now, eventually, these things go out and they're able to surround clay particles, and the metal ions that pull together the different components of those organic molecules are the same systems that will pull organic matter, humus, that can completely surround a clay particle. Well, when it does that, suddenly when you look at it, you're not seeing the clay particle inside, you're seeing the organic matter on the outside. Well, the clay particles, the attractive cements that they have to form aggregates are water soluble. So every time the clay gets wet, it falls apart. But once you have these molecules holding them together and coating them, then this forms a waterproof sticky cement that will hold soil together even when it's very wet. Okay, so here's kind of an illustration. Here you have the organic humates out here, and then you have water molecules, and their hydrogen atoms on the water molecules have a very weak bond that actually can help hold the organic humic acids onto the clay particles. Okay, well, you can imagine you have this little string of this stuff out there in the soil, and an earthworm comes along and it just gobbles this up. And once it's inside its digestive system, this rolls around and around and around. And in each one of those little individual strands of clay humus complex wrap around and around and around each other. And they pull in silt-sized particles in with them. And pretty soon, where you used to have silts and clays, you now have small sand-sized particles that are actually the accumulation of clays and silts. And that's what these systems begin to form up, creating this ever more stable soil. And this is what you end up with. A nice soil. See how brown it is in there? You see it's wet, but it hasn't fallen apart. And that's because it's being held by this beautiful brown, sticky, organic cement. So, and it sort of begs the question, is there any way that you can grow food, grow crops, and still keep these processes intact and going? Well, of course, no-till agriculture is a great place to start. And using no-till drills and growing cover crops and all the things that are traditionally done in no-till agriculture is a great step forward. But this is an example of a system where you actually keep everything intact. In this case, we dig trenches. They're, in our case, about 4 feet deep and 18 inches wide. And then we have 4 foot wide growing beds. And then every time we grow a cover crop or a vegetable crop, we harvest absolutely all of the biomass from the spent crop or the cover crop, and we bring that into the trench. And then we bring that material in and we cover it with wood chips in order to put and the wood chips very, very, very quickly completely fill with fungal hyphae, and that makes an airproof layer, an occlusive layer between where the detritus is breaking down, using those detritivores to break down the organic matter. And then it has absolutely no choice. It's already underground in the soil, ready to go. And then the walls of these trenches very, very quickly become completely filled with detritivores, notably worms. And it'll be absolutely shoulders thick with worms. And what they do is they take the nutrients that are developed out of the detritosphere, they ingest them, they immediately expel them into the porosphere where they're stored. And then the rhizosphere stays intact, completely enclosed in the other layers. And so you're now using this, the soil spheres to cycle nutrients into the rhizosphere. The plant grows, takes the nutrients, puts them up into the part of the plant that's above ground. You harvest that, put it back in the ground. All the roots remain undisturbed in the soil around it. They decompose. And since all this time you have all those guys in the on the aggregates generating brand new nutrients, what happens is that nutrients over the course of time accumulate in the rhizosphere and make it richer and richer and richer every single season. And an important part of this is that the carbon that is generated, all those fragments that come out from the chitin and the lignin and the subrin and so forth, since they're kept away from the atmosphere, as those as the biomass decomposes, all of the moisture that was in the biomass is drawn out into the soil. And since freshly cut plant material is probably 60% moisture, that moisture goes directly into the soil where it can be utilized. And all the nutrients, the calcium, magnesium, manganese, molybdenum, all of them are digested here, absorbed into the drillosphere and into the porosphere and brought right into the rhizosphere. And the only thing that's left are those carbon compounds. And what they do is they accumulate in the bottom of the trench. And we put a layer of more resistant materials. In hugelkultur, you use wood and so forth. And what that does is it puts a barrier between this carbon and the soil below it. And that allows the carbon to accumulate and not go through a further decomposition or oxidation. And so just exactly like happens in a coal swamp, you have organic material. It goes into, in this case, in a swamp, it goes underwater so that the atmosphere can't get to it. And all the critters that are in the decomposition in the detritosphere down below in the swamp layer are pulling out the nutrients. And in a coal swamp, these nutrients are actually utilized. They don't accumulate because coal swamps have a lot of biological activity and a lot of leaching of the nutrients that are in that material. And so the only thing that's left is the carbon that was left in the biomass. And it begins to accumulate and turns into coal. And coal, just like rocks, are composed of a whole bunch of different kind of minerals. And the way you come up with a name for a rock is by looking at the minerals and calling it a granite or a basalt, depending on the size and characteristics of the minerals. In the same way, coals are made up of all different kinds of macerals, analogous to minerals. And when they go prospecting for coal, they will look and determine what these macerals are, and they can determine the kind of coal that it is by what the macerals were. And you see that this group of coals are generated from fungal decomposition and charcoal fires. And these kinds comes from spores and leaf cuticles. And these come from humus, from the woody parts, the lignins and subrins and cutins and all of those. And so all we're doing in our process here is just emulating this process. Now, the beautiful thing about this process is that once, and this material actually forms down to the bottom of our trenches. If you dig it up, you'll see this layer of black material. And we've done some of the rudimentary chemical tests in this, and the material that accumulates in the trenches acts chemically exactly like this material in all the tests that we've applied. And so you can see these layer after layer after layer after layer. Well, back wherever this piece of leonardite formed, probably back in the Eocene from these gigantic forests, it laid down layer upon layer upon layer over millennia, or eons perhaps. And we're just doing exactly the same process. Now, the beautiful thing about carbon, once it makes it into this form, is the only way that carbon can make it back into the atmosphere is if you burn it. You can leave this on the surface, you can grind it up, you can put it in a bag and sell it. You can mix it up in solution in a jug and sell it. And as long as nobody sets it on fire at any stage of the game and simply puts it back into their soil, the carbon in this stays in this form forever. And that is the only way that I've seen that you can actually sequester carbon in an agricultural setting. And there are, if you find, if you're growing specific crops that are very, very, very high in lignin and suberin and so forth, the more of this material you'll accumulate. And if you look at the volume and the mass of biomass that you can put into one of these systems, you can accumulate a tremendous amount, tons of sequestered carbon in these systems. So the only thing that you have to do to make sure that this, first of all, that the chemical process occurs and that it stays in this form, is you have to keep it away from soil, because soil will oxidize this on its way to becoming coal minerals. You have to keep it away from water, because water will inhibit the formation of these minerals. In coal swamps, at the bottom of the coal swamp, the first thing that accumulates is peat, which can occur underneath water. But in order for that peat to then turn into coal, the water has to go away, because the water gets in, interferes with these layers accumulating. And so, pardon me. And so if you keep this material away from water, away from the atmosphere, and away from soil, this material will accumulate all on its own. The formation of coal is a biological process. The formation of the process that turns this lignite into the coal that you use as a combustion source, that's a geological process. But the initial laying down and accumulation of the carbon is a biological process. So otherwise, if you're not accumulating carbon in your trenches, what other ways are great for getting organic matter and then carbon into the soil? Well, composting, you're just doing the same thing we're doing in the trenches, except that you have to collect things and carry it off and compost it. And in the process, you'll lose most of the nitrogen and probably 50% of the carbon. Then you have to carry it back and distribute it in wherever you're doing your agriculture. The nice thing about the trenches is that since we cut off the biomass and put it in the trench right next to where the biomass grew, it's only like 2 feet away from where it started out. So you know that the nutrients that came out of a soil are going right back into the soil, right where they came from. Now, another really good source of organic material for soils are worm castings, probably the very, very best, because worm castings hold together well. They form very good long-term organic molecules in the soil, so they don't decompose quickly, and they provide a lot of enzymes and a lot of calcium, because worms actually generate calcium carbonate in their processes. So that's really hard to beat. And then mulching protects the surface of the soil. It keeps moisture in the soil. It provides a layer for all those detritivores to hide under and decompose the mulch and turn it into soil. And of course, it inhibits weed growth as well. And then probably the most fast-acting organic substance amendment, if you will, that you can add to soil is humic acid. You can make the dilution of humic acid, spray it directly on the soil, and almost watch in real time as those humic acid molecules seep down into the clays and silts and transform it almost on contact. The only downside about humic acid is virtually all of it that you can purchase comes from mines, most of them in either Germany or in, I think it's North Dakota, in one of the Dakotas, I think it's North Dakota. And the environmental impact of mining that humic acid is pretty substantial. So, you know, that's the downside of it, but it's incredibly fast acting. So that pretty well covers all the points that I wanted to get to in this. We have some great resources that I'd be happy to share and perhaps generate a PDF to go along with this presentation. But that, I think, really covers how you can look at soil health and some of the systems that are really necessary to be there in order to allow biology to do all of the hard work for you. Because honestly, once you get these systems going and activated and getting the soil biology to do all the work and simply cycling the nutrients back into the soil where they came from, the only thing you have to do in producing food or agricultural products is plant and harvest and make sure that you return the biomass back to the soil. And that really, really makes it a whole lot simpler to do gardening or farming. And you just let the microbes really do all the work. How about that?
Right. Terrific, Scott. Thank you. Super inspirational. And I'm always glad to see it again. I'm glad we've been able to capture this for future classes. And thanks so much. I will turn off the recording.
Thank you.